Hello everyone, welcome to Science Tutorial. Today we are going to perform a paper chromatography method. In this method, we required petroleum ether, 60 to 80 degrees Celsius, then cotton, and then test tube stand, leaves of spinach, mottle and pistal, petridish, and capillary. Okay, this is the requirement of paper chromatography method. So first of all, we take leaves of spinach. I already taken an abstract of leaves. So we need to prepare a abstract from the leaves. With the help of mortar and pestle, we need to crush the leaves and collect the abstract from the leaves. And this abstract already taken in Petridish. So main principle of this method is how to separate the pigment from the plant. We all know in plants some pigments are present in the plant like chlorophyll A, chlorophyll B, xanthophyll and carotene. Okay, these are the pigments which are present in plant, and this pigment have different type of color. Like chlorophyll have a green in color, xanthophyll yellow in color, carotenoid which is present orange in color. Most of the part of plant, especially leaves, contain some combination of these three pigment. It is possible to separate this pigment from each other using this paper chromatography method. So we take a Wattman paper number 1, like this strip we will take, and then after, with the help of pencil and scale, we mark the pointer like this. With the help of scale and pencil, we indicate the pointer which we put the abstract of the leaves like this. You can see here. Okay, like this. Draw a line by pencil. In the strip of the middle, we can draw vertical line like this. Okay, and then after, we cut the chromatography paper in a pointer view. This type of cutting will help the Wattman paper easily dipped in the solution, and the solvent can easily moved in upward direction. Okay, like this you can see here. So this type of strip ready for the practical. So we can put the first drop of the middle of the strip with the help of capillary. We can put the first drop in the middle of this Wattman paper, which we have to indicate with the pencil. A drop of abstract we will put here and dry it for some minutes, and then other drop we will put. Okay, and then we need one test tube, and the test tube filled with petroleum ether like this. We take a small amount of petroleum ether, and then after, the strip is ready to dip into the solution like this. So we can easily perform this type of separation from the plant. Different pigments are separated by this method from the plant. Okay, and then after, close the test tube with the help of the cotton like this. You can see here. By this method, you will easily separate the pigment from the spinach leaves. The key to identify the different pigment in the leaf is measuring how far the pigment has traveled from its starting point. Be very precise in your measurements. You will also calculate the RF value for each pigment. This factor is calculated by comparing the distance the solvent has moved with the distance the pigment has moved. Okay, this practical is done by this method with different type of pigment separated by this method. So you can easily calculate the RF value. RF means retention factor, जिसका फॉर्मूला है distance traveled by the substance upon distance traveled by the solvent front. So with the help of this formula you can easily calculate the solvent which are traveled from the Whatman paper. Okay, this type of color you can see on this strip: chlorophyll A, B, xanthophyll and carotene. If this information helpful in your study, then like, share and subscribe it.